Bismillah ar-Rahman ar-Rahim. This is a rapid revision for the digestive glands. After seeing the original presentations of these glands, liver, pancreas, salivary glands, now you can assess your knowledge by this rapid revision. Now, this is the first slide. Identify the type of the acini. As you see here, we have these acini with the narrow lumen, and the cells are more or less basophilic and granular, so these are serous acini. So this the serous acini. Mention two visible criteria. The two visible criteria: number one, the very narrow lumen, and the cells are basophilic with granular cytoplasm. Then the yellow arrow points to this duct. Of course, it is a wide lumen, so it is a duct, and it is inside the lobule, so it is an intralobular duct. And as you see, it is formed of cuboidal cells or low-columnar cell, and it is, of course, the striated duct. So yellow arrow points to the striated duct. Name the cell pointed by the red arrow. This is, of course, the fat cells, which are present in huge numbers in this gland. Of course, you know this gland is the parotid gland, because here it is purely serous acini. Now, the second slide. The red arrow points to, of course, these are serous acini, typically basophilic granular cytoplasm and very narrow lumen. Black to the fat cell, typical vacuolated due to dissolving of the fat during preparation, and we have a signet ring peripheral nucleus. And the yellow arrow points to the striated duct, which is characterized by being lined by cuboidal or low-columnar. Cuboidal, we have here the rounded nuclei, but the height is very much enlarged than the typical cubicle, so it is cuboidal, or sometimes you can say that it is pyramidal sometimes. Here we have the striated duct. The green arrow here, also we have here the striated duct. The green arrow here points to what? It points to an acinus which is characterized by having a wide lumen and lined by cells with vacuolated cytoplasm. It is a vacuolated cytoplasm, and look carefully for the crescent. This we have a crescent of a basophilic cells, so this is a serous demilune. Here it is present also, here it is present, and here, and so these are the mucoserous acini. So this is a mucoserous acinus showing mucus cells and a serous demilune, and this is the striated duct. Here, identify the gland. You should look carefully for the type of the acini. I can see here serous acini, basophilic, narrow lumen, and we have also here vacuolated cells. Here we have a vacuolated cells, meaning that it is a mucus cells. And here we have also serous demilune. So we have here three types of acini. We have a serous, we have a mucus, and we have mucoserous. So it is the submandibular gland. Human, of course. And here we have the yellow arrow. The yellow arrow points, of course, to this duct, which is lined by the cuboidal cells, and this is the striated duct or intralobular duct. And the black arrow here points to these acini formed of basophilic cells, narrow lumen. These are the serous acini. The yellow star here indicates a typical mucus acinus formed of the cells which have a vacuolated cytoplasm and basal flattened nucleus, and they are low cuboidal cells or low cubical cells here, and with the basal flattened nuclei, vacuolated cytoplasm. I cannot see here any serous demilune, so they are the mucus acini. The red arrow here points to these basophilic cells on the top of this mucus acinus, so it is the serous diminution of the mucoserous acinus. Here we have slide number 4, which is for the pancreas. The field shows the parenchyma of the pancreas. How we know that it is a pancreas? We have here an islet which is pale stained among the darkly stained pancreatic acini, which are characterized by having these cells, pyramidal cells with basal rounded nuclei and apical acidophilic cytoplasm, and the basal part is basophilic, so it shows two colors, the red at the apex and blue at the base. So these are the pancreatic acini, and this is the islet of Langerhans. Identify two components of the glands. Of course, the islet of Langerhans is this structure marked by the letter A, which is faintly stained, not surrounded by any capsule, formed of cells scattered, not arranged in the form of acini. And they secrete the pancreatic hormones as insulin and glucagon. The pancreatic acini, rosette-shaped, formed of these pyramidal cells with basal rounded nuclei, and they have basal basophilic cytoplasm and apical acidophilic granular cytoplasm. Here we have another slide. This is a section of the pancreatic showing pancreatic acinus. What is in this center? It is a centroacinous cells, which is the lining of the intercalated duct. And here we have the yellow arrow points to the zymogen granules present in the lining cells of the pancreatic acinus. And now this is the picture for the liver. As you can see, this is the central vein and the cords radiating from it formed of hepatocytes. The black arrows here, they are pointing for the blood sinusoids. The black arrows for the blood sinusoid. As you see, you can see here these the blood elements can be seen in these spaces between the cords of hepatocytes. And then the yellow arrow points to the nucleus of hepatocyte, and the arrowhead points to the central vein. Now, this is a section in the liver. As you see, we can see this central vein, and here the blood sinusoids passing their blood to the central vein, and you can see these cells present hanging in the wall of the blood sinusoids, and here we can see the blood, and this is a neutrophil here and lymphocyte present in the blood of the blood sinusoid. So this is section in the liver. The black arrows points to the von Kupffer cells, of course, here in the wall of the blood sinusoids, stained by any vital stain, of course, like India ink. And here we have the yellow star indicates the central vein, and the red arrow points to the blood sinusoid. Very nice picture for the blood sinusoid, showing the white BCs inside. Here, this is a section showing a structure with a lumen and a folded mucosa like this, and this is, of course, this is a section in the gallbladder. The lining epithelium here, as you see, it is simple one layer columnar epithelium, and this blue star indicating the musculosa layer of the gallbladder. Then we have this pattern connecting between three central veins. It is a triangle drawn between the three central veins. This is the portal lobule, and it includes all the hepatocytes that drain their bile into a common bile duct in the portal tract here. Then this is a picture, of course, for the portal tract present at the corners of the classic hepatic lobule, and it shows what? The yellow arrow. The yellow arrow here points to this bile duct because it is lined by simple cubical epithelium with typical central rounded nuclei, and the black arrow here, it points to the blood vessel, which is a branch of the hepatic artery. And here we have these arrowheads pointing to the blood vessel, which is a branch of the hepatic portal vein. How do you differentiate between both of them? Here we have the wall is thicker, and here the wall is very thin, a narrow lumen, wide lumen, and of course they are typically different from the bile duct from the lining. Here the lining in both of these two is the flat squamous epithelium, but here the lining is a simple cubical epithelium. Thank you. Best wishes for all.